Magandang umaga sa lahat. Welcome to Connected to the Wild Biodiversity Research Series. We are now on the second episode of a series of webinars featuring different research studies on Philippine biodiversity. These studies were funded and supported by the United States Agency for International Development of USAID, or USAID, under its Protect Wildlife project. Before we proceed to the webinar proper, we'd like to remind everyone of a few house rules. So kindly mute your mic and turn off your video during the webinar. I-turn on lang natin ang mga ito kapag ina-acknowledge po namin kayo during the open forum later on. We have two back-to-back presentations this morning. So if you have questions during these presentations, you can post this on the Zoom chat. I-click lang ang chat button sa ibaba ng inyong Zoom screen. And pwede kayo doon mag-post ng inyong mga tanong. We'll be reading your questions during the open forum right after both presentations. If you have questions during the open forum and you want to join in the discussion, click the participants button sa inyong Zoom screen and then click the raise hand button so we can acknowledge you. So those are our simple house rules. By the way, I am Evangeline Miklat, Senior Specialist for Science, Technology, Innovation, and Partnerships at Protect Wildlife Project. Again, welcome po sa lahat joining us for this webinar here at Zoom. We also want to welcome those who are joining this webinar via live stream at USAID Protect Wildlife Facebook group. If you are not yet a member of this exclusive group of Protect Wildlife partners and conservation enthusiasts, i-search lamang po ang USAID Protect Wildlife sa Facebook and join the group. At this point, we would like to acknowledge our partners, the Department of Environment and Natural Resources. We have here the Biodiversity Management Bureau, particularly the Wildlife Resources Division, and also the Caves and Wildlife and Other Ecosystems Division. We also have the Forest Management Bureau and the Ecosystems Research Development Bureau. We are also joined by higher education institutions. We have graduate students who are science educators from the Ateneo de Manila University, the Nueva Vizcaya State University College of Forestry and Center for Environmental Resources Management. We also have the University of Santo Tomas. I also acknowledge the presence of Nueva Vizcaya, Penro, and the Nueva Vizcaya Tourism. And we are also joined by USAID and our Chief of Party of Protect Wildlife. So to begin with, I would like to share a little bit about the USAID Protect Wildlife project. It has been under implementation since 2016, and we have been working with the DNR and government agencies, colleges and universities, non-government organizations, and civil society to accelerate partnerships and opportunities for science-based conservation efforts. Under this project, we support researches which generate scientifically rigorous evidence and knowledge for practical application in biodiversity conservation focused on endangered and threatened wildlife and their critical habitats. Our researches also support protected area and ecosystem management and protection of other key conservation and management areas in the country. We also support research that enhance education and outreach programs, science-based policy formulation, development of technologies for resource-based livelihoods, and improvement of wildlife and environmental law enforcement. So hindi na po natin papatagalin. We begin with our back-to-back presentations this morning. Today we have Inigo Luis del Prado of the University of Santo Tomas presenting his research on the Philippine pit viper, and Dr. Jason Carranza of Nueva Vizcaya State University presenting his research on the Capisaan Cave System. Our first presenter is Mr. Inigo del Prado, an MS Biological Sciences student of the University of Santo Tomas. His study is on the identification of the cryptic species of Philippine pit viper in the Luzon biogeographic region with the use of molecular or mitochondrial analysis. Proper identification of a species is important to determine appropriate conservation measures because we cannot protect what we do not know. His research is not only important to taxonomy, but also to medical application, because if the identification is ambiguous, it poses a problem in the snake bite treatment. So now I give the floor to Mr. De Prado.
Thank you, Ma'am Vanji. Good morning. Uh, First off, I would like to thank all of you for joining us here today. And of course, a big thanks to USAID for organizing this event and for the Protect Wildlife program. Their generosity and support helped make this study possible. I would also like to thank the Department of Science and Technology, the Department of Environment and Natural Resources, the National Museum of the Philippines, UP Diliman, and of course, the University of Santo Tomas. So to give everyone a heads up, the study is a bit technical and I promise to do my best to explain everything well. So with that, yeah. So I'm sure most of us here know all too well and can vouch for the recognition of the Philippines as a mega diverse country with a very high concentration of endemic biodiversity. So these are plant and animal species found nowhere else in the world. This rich biodiversity is partially explained by an equally rich geographic history and unique topography. Yeah. So following the numerous biodiversity studies done in the Philippines, the country's Pleistocene history helped reveal unique patterns in biodiversity, where such as lower sea levels during the glacial periods repeatedly connected adjacent islands into larger land masses called Pleistocene aggregate island complexes, shown here in gray. Uh, this allowed animals to cross onto nearby islands. So five major paeks were formed: Luzon, Mindoro, Palawan, Negros Panay, and Mindanao. So each of these paeks contain unique animal species and are now considered uh, primary regions of biodiversity, hosting distinct sets of wildlife. Yeah. So additionally, the unique topography of each landmass has helped explain intra-island biodiversity. So mountain ranges, such as those found in Luzon, have been found to contain localized microendemism, species found only on those specific mountains or islands, further identifying them as biogeographic subregions. Now, unfortunately, the Philippines is also recognized as a global biodiversity hotspot with an increasing threat of species extinction. So like the virus threatening the human population today, habitat loss, climate change, introduction of invasive alien species, and the emergence of disease have long been propelling plants and animals toward extinction. This puts us in a race to identify and underscore the importance of each and every species to take the appropriate conservation steps. In biodiversity studies, however, naming one species from another is not always easy. So, a lot of species have been traditionally described solely on the basis of looks, measurements, and unique features. However, the presence of two or more species that are superficially indistinguishable and existing in separate regions has long been a cause of confusion for taxonomists. So these so-called cryptic diversities or cryptic species have been discovered in widespread groups that are polymorphic, meaning they display varying colors, uh, where more recent studies, such as on Philippine varanus or mga bayawak, have highlighted the key role of using molecular techniques in finding these hidden species, coupled with a closer look into their morphology, ecology, and distribution. Now, another species notorious for cryptic speciation is in Southeast Asia is the genus Trimeresurus, or the Asian green pit vipers. Now, the genus belongs to the subfamily Crotalinae, or pit viper, and are the most diverse group of venomous snakes in tropical Asia. Uh, they are also the most commonly encountered venomous snakes in Southeast Asia. Members are very difficult to classify due to their wide distribution range and very similar anatomy, and have since been split into genera and several subgenera. There are currently around 50 species of these green pit vipers, with numbers still increasing as cryptic species are revealed through re-examination using molecular techniques. Now, going back to the Philippines, we are lucky to share this archipelago with 3 species of Trimerosaurus. These Philippine species are allopathic, meaning they are found in separate, non-overlapping regions, with Trimerosaurus schultzei, endemic to the islands of Palawan, Trimerosaurus macgregori, endemic to the Batanes group, and Trimerosaurus flavomaculatus, found throughout the country. All 3 species belong to the subgenus Parayas, a relatively understudied group of Indomalayan pit vipers. Of the 3, Trimerosaurus flavomaculatus is the most widespread, so it occurs in almost all major Philippine islands except for Palawan. This earned it the name Philippine pit viper. The species is a lowland forest ambush predator known to display a variety of color morphs. Its wide distribution and polymorphism has largely led to a complicated taxonomic history. So, the species was originally described by Gray in 1842 as 3 separate species, which were later found to be one and the same. It was later, again in 1964, split into 3 subspecies, where Trimerosaurus flavomaculatus macgregori was later reconsidered as a separate species based on distinct coloration patterns found only in the Batanes group of islands, and later found that there's a difference in their skull shape. The other, Trimerosaurus flavomaculatus haleus, was found to be a color morph found only on the island of Polillo, which was later also found in mainland Luzon and was no longer recognized as a valid subspecies. Now, from the complex, only Trimerosaurus macgregori and Trimerosaurus flavomaculatus are presently recognized as valid species. Now, given the complex patterns of biogeography in the Philippines, the variety of color morphs and wide distribution range of flavomaculatus, we found Trimerosaurus flavomaculatus an ideal model to study for cryptic diversities. So, as an initial step in unraveling the secrets of the Philippine pit viper, here we use DNA to establish the evolutionary relationships of the species with comparison of varying measurements to investigate for cryptic diversity among populations in Luzon. So, good morning. I am Inigo del Prado from the University of Santo Tomas, and that was my very long intro to the study. So, with the goal of covering the entire known species range and including samples from all pikes, here we first focused on Luzon. We aim to document and describe all morphs in the region, to infer the phylogeny using 4 mitochondrial markers, and to investigate if cryptic diversity is found within the region. So to accomplish these objectives, we targeted sampling sites covering the 5 known biogeographic subregions in Luzon: the Cordilleras, Sierra Madre Mountain Range, Zambales Mountain Range, South Central Luzon, and the Bicol Peninsula. And with the necessary collection and transport permits, we spent 5 to 12 nights in each site doing both day and night opportunistic sampling, visiting streams, river habitats, and those recommended by our guides. We used snake tongs, rakes, or improvised snake hooks, or aka kawayan, to capture and place snakes in specimen bags. Following snake preservation protocols, snakes were prepped, dissected for liver and muscle tissue for DNA analysis, and preserved. Now, these voucher specimens are deposited in the National Museum of the Philippines. And to compare variations in the species features, we followed standard morphological characters used in separating taxa for the genus Trimerosaurus, where a total of 33 characters were considered, including coloration and pattern. So, for the molecular part of the study, from the liver and muscle samples, DNA was then extracted, amplified, and purified. These samples were sent to Macrogen, South Korea, for sequencing. And finally, these sequences were used to reconstruct a phylogenetic tree, which shows us the relationships among included samples based on similarities and differences in their DNA. So this is an ingenious tool found to be useful in establishing species boundaries. Now, taxon sampling resulted in 18 adult voucher specimens of Tremerosaurus flavomoculatus. These individuals were found at night between 8 p.m. to 2 a.m. in riparian habitats of second growth forests, around 300 to 700 meters above sea level. Individuals were found either actively moving through the brush or leaf litter, perched as high up as 5.5 meters on tree branches directly above streams, or in an ambush position while perched on roots, boulders, or lower branches around streams. So additional specimens were borrowed from the collection of the National Museum of the Philippines and from the Biodiversity Research Laboratory of the UP Diliman Institute of Biology for a total of 28 individuals, representing all subregions, including regions outside Luzon. Now, morphological variation among all individuals agree with published descriptions of the species by Leviton 1964 and Gumprecht et al. 2004. Among these samples gathered, sexual differences are observed in overall total lengths, with females evidently growing much larger than males, as seen on the figure here. Take note, these are both adult specimens, where the one above is a female and below is a male. Additionally, despite being shorter overall, males have larger tail to total length ratios with a higher number of scales. And yes, I counted all these scales manually. Now, compared with other species of Trimerosaurus, scale counts and measurements are very similar, as expected for known cryptic groups. Yeah. So these species, however, differ greatly in coloration and biogeographic distribution. So the similarity in scale count is explained by Sanders et al. As adaptive evolution linked to similar behavioral and environmental patterns. As again, these are all Southeast Asian species. Now, from the sampled Luzon populations, color and pattern was evidently highly variable. From these, 4 morphotypes occurring in sympatric or overlapping distribution were observed. 3 of the 4 correspond to the 3 species originally described by Gray in 1842, and so we named each morphotype accordingly. So the first one we call the true flavomorph, described by Gray as Megaera flavomoculatus, having green bodies and yellow spots along the sides of the body. The second species, described by Gray, is Megaera variegata, a green snake with prominent reddish-brown dorsal crossbands. Here we name specimens of this morph as the variegata morph. So compared to the true flavomorph, these snakes have an intense reddish-brown pattern on the back, where the head also has distinct markings and the body has 2 to 3 scale-wide crossbands. So the sides of the body display alternating yellow and reddish-brown spots. Now, the last species described by Gray was Megaera ornata, a green snake with irregular brown dorsal spots and white over brown dorsal side spots. Snakes displaying this morph are named the ornata green morph, having reddish-brown spots on the backs and having a series of white spots over a red line on the sides of the body. Yeah. Now, interestingly, Gray did not describe a red morph for the species. However, a red flavomoculatus morph has been well documented. We were able to find adult individuals of this morph for both male and female and call it the ornata red morph. Now, ornata reds are very similar to ornata greens regarding pattern, but instead have a reddish-brown dorsal and ventral base color. Now, additionally, a remarkable observation is that Mindanao populations appear to have a set of morphotypes likely to be unique to the faunal region. So these photos show 3 morphotypes from Mindanao. So this distinction between morphotypes of Luzon and Mindanao has already been mentioned by several authors who have suggested the need to re-evaluate the taxonomy of Tremerosaurus flavomoculatus, considering Mindanao, specimens of Mindanao as possibly a separate species. So moving on to the molecular part of the study, in reconstructing the species phylogeny, we produced 78 sequences from 21 individuals of Trimerosaurus flavomaculatus from 6 populations using 4 mitochondrial gene regions. So basically, this will increase the total number of sequences for the species available to the public in online databases from 4, which are currently published, to 82. So once uploaded, these sequences can be used by other researchers in various studies. Now, these sequences were used to produce well-supported trees for both maximum likelihood and Bayesian inference analysis with very similar topography. The trees form a strongly supported monophyletic group for the subgenus Parias, agreeing with previous studies. This basically means that the inferred relationships for our chosen group, which is Parias, is resolved with all members sharing a common ancestor, as represented by the white bar. The analysis, however, revealed an unresolved complex for 2 Philippine species, as shown by the red bar. Yeah. Zooming in on the complex, Trimerosaurus flavomaculatus was found to be paraphyletic or unresolved, where 3 lineages were formed. So, Trimerosaurus macgregori in yellow and Trimerosaurus flavomaculatus from Luzon and Mindoro in blue, which formed a sister relationship. The single flavomaculatus from Mindanao diverged earlier, forming a strongly supported relationship with the rest of the group. So ideally, to say the relationships are resolved, a group must contain all members of the same species under one common ancestor. Here, the insertion of Trimerosaurus macgregori within the flavomaculatus group warrants further investigation of their taxonomy. Now, within the Luzon clade, the inferred trees show 2 poorly supported lineages for Flavomaculatus of Luzon, splitting populations between northern and southern parts of the island. The 4 markers were further able to split individuals into the 5 subregions. However, based on morphological comparisons and genetic distances, there appears to be insufficient evidence to suggest cryptic speciation within Luzon. So, going back, the placement of Trimerosaurus macgregori within the clade and the paraphyly of Trimerosaurus flavomaculatus suggests the possibility of a cryptic diversity within the complex. So this allows us to consider 2 possible resolutions. The conservative approach would be to recognize the 3 lineages as a single species, Trimerosaurus flavomaculatus. This, however, would disregard morphological, ecological, and biogeographic evidence supporting the distinction of Trimerosaurus macgregori, where a more ideal proposal would be to consider all 3 as separately evolving isolated lineages, recognizing the validity of Trimerosaurus macgregori, and treating Flavomaculatus of Luzon and Flavomaculatus of Mindanao as separate species, for which the name Trimerosaurus schadenbergii is already available. Now, to summarize, we documented 4 widely occurring morphotypes within Luzon, where 3 correspond to the 3 species originally described by Gray in 1842. So, results of the phylogenetic analysis confirms the placement of Trimerosaurus flavomaculatus within the monophyletic subgenus Parias. However, Flavomaculatus is found to be paraphyletic, and an unresolved complex is revealed where 3 lineages are evident. So the paraphyly of Flavomaculatus plus the presence of allopatric sets of color and pattern morphs suggests the possibility of a cryptic diversity between biogeographic regions. However, as this is an initial step in hopefully including samples from all over the Philippines, hopefully, and due to the limited samples available, a formal decision is yet to be made, and the phylogeny of Trimerosaurus flavomaculatus remains unresolved. So here we provide the first attempt at a deeper investigation of the phylogeny of Trimerosaurus flavomaculatus. So these findings further highlight the need for taxonomic reevaluation, where the treatment of the 3 lineages as separate species would resolve the problematic clade without overlooking the distinctness of Trimerosaurus macgregori. With this, we recommend a more extensive sampling of Trimerosaurus macgregori and Flavomaculatus throughout the entire range of the species. Having enough samples representing different biogeographic regions would allow the use of quantitative morphological methods to help in species delineation. And with that, I hope my short presentation has introduced you to the colorful world of pit vipers. So we still have much to learn about our country's great biodiversity. And with the help of our generous partners, we're slowly creeping towards unraveling the Philippines through diversity. Yeah. So again, the big thanks to USAID for making this study possible. Also, a big thanks to the following people for sharing their photos. And thank you all for listening.
Maraming salamat, Inigo.
Thank you.
If you have questions for Inigo, please type them first on the Zoom chat, and we'll read your questions during the open forum right after the second presentation. So while we wait for our second presentation to get ready at this point, we would like to invite everyone for a quick photo op. We request everyone to turn on their cameras and then look towards the camera and smile. And I will give you a cue at the count of 3. So we'll have Protect Wildlife staff prepare for it. Thank you.
How do I start? Oh.
So while our staff are getting ready, I would like to acknowledge the presence of the Association of Tour Guides of Negros from Bacolod City. Thank you for joining us. So do we have everyone on camera? Okay, on the count of 3. 1, 2, 3. Thank you, everyone. So again, we will request you to turn off your cameras and also your microphone. And we will be ready for the second presentation. Our second presenter is Dr. Jason Carranza. And yes, he has successfully completed the requirements of PhD in environmental science at the University of the Philippines, Los Baños. Dr. Carranza is an assistant professor at the Nueva Vizcaya State University. He will present to us the recreational value, sensitivity, and disturbance of Capisaan Cave System in Nueva Vizcaya. He will take us to a tour of the cave by presenting its biodiversity features, significance to ecotourism, and why it is important to conserve the cave and cave system while deriving economic benefits from them. His findings are valuable inputs to considering options for ecotourism, managing tourist activities, development of science-based materials for education and community awareness, and landscape planning and management. Interestingly, his study employed methodologies that can be replicated in the assessment of other caves and cave systems in the Philippines. So now I give the floor to Dr. Carranza.
So thank you very much for a very significant attendance for this morning. I think we're almost 100. It's 90. That's very amazing. So I would like to present to you my dissertation, which we conducted at the Capisaan Cave System, in which we tried to see or evaluate the recreational value, sensitivity, and disturbance of the Capisaan Cave System. And a little heads up, there will be a quick rundown of slides for the interest of presenting as much as all the results that we gathered for a very limited time that we have. So For this research, we all know that protection of ecosystems is a vital component for sustainable development. And karst landscapes are very special since we have cave systems which offer huge ecotourism potential. But then, more often, they are subjected to deforestation, overexploitation, and sometimes mismanagement. And of course, the recreational uses of caves is affected by the quality of these derived services and resources that we see in caves, which is particularly the richness and vulnerability of these resources and the level of disturbance it sustained. So knowing a cave's level of sensitivity and disturbance is vital for the efficient creation and execution of protection and management policies, especially at each local level, since each cave is different from one another. And different parts of caves, especially of long caves, have different physical formations that have varying levels of vulnerability. And of course, even the sensitivity to disturbance. So how to correctly evaluate the relative disturbance and vulnerability of these passages is the answer that we tried to, or the question that we tried to answer in this research. So the objectives of this research is to explore the value of the recreation services provided by the Capisaan Cave System and to assess the overall socio-ecological condition of the cave by looking at the economic and the biological and social components affecting the cave system using a sensitivity and disturbance index. So this is the conceptual framework of this research. Particularly what we tried to understand is the recreational value, the sensitivity score and disturbance score, which would describe the current socio-ecological condition of the cave, which would then give information or implication to management priorities and decision making for the efficient caretaking of these resources. So this is the site of Capisaan Cave, which is in Nueva Vizcaya, particularly in the town of Kasibu, which is the fifth longest cave system in the country with almost about 4.2 kilometers total passage length and known to have nine known entrances. So it is the area of the karst landscape is about 1,515, so it's not that big, and has an elevation of 700 to 1,200 meters above sea level. So for the methodologies and the results, first we tried to identify the biophysical or describe the biophysical, social, economic, and political status of Capisaan Cave, in which we tried to review secondary data, direct observation, interviews, and focus group discussions, including map overlay analysis to be familiar of what is the current condition of this area. So the key findings is that since it has been opened for tourism in 1992, including its exploration and initial development, has resulted to a lot of infrastructural changes. But then at the moment, it has now a forest land use plan, which is covered by the municipality and has also a recognized cave and tour guide association. And it is now currently managed through a multi-stakeholder approach between government and non-government institutions. And lately, it is officially declared as a local conserved area of the province of Nueva Vizcaya. And also, ecotourism in the area has improved the economic condition of the community, as shown by those increased guiding jobs and other livelihood. But then, included in this development is the changes in land use and land cover of Capisaan, which is very evident with the increase of build-up and agricultural areas over time. So let's proceed to the estimation of the recreational value of the caving activity in Capisaan, which is done primarily by the use of the zonal travel cost method. wherein we tried to interview visitors through a supervised self-administered survey for 10 months. And this happens from July 2018 to April 2019. Uh, we also consulted the logbook of the Capisaan Cave Information Center, which is present on site, to know the use value or the consumer surplus plus the travel cost that the actual number of visitors incur in visiting the site. So in the zonal travel cost method, we tried to make zonal boundaries, and these are the 7 boundaries. The Capisaan Cave System has an average visitation rate for the past 3 years of more or less 4,500 visitors a year, and increasing yearly. And from the survey, we have 429 respondents. So for the zonal 3-year average visitation rate of Capisaan, it is presented here through the different from the different zones. Uh, uh, for the consumer surplus estimate, we try to use the 1,000 visit per 1,000 population data and using the actual visit. And we try to interpolate the data that we have using different data transformation, the linear model, the semi-log model, and the double log model. And we can see from this table that the semi-log model and the double log model has provided us a good estimate of the consumer surplus of Capisaan Cave System caving activity. So from this, we were able to identify that the Capisaan Cave recreational value ranges between 1.7 million to 5.7 million, or for the total access value of 8.3 to 12.3 million a year. So that is for the economic. Now let's proceed to the biophysical. So we did cave mapping, cave survey, and limestone measurements also in the area. And using a freeware, which is the WinCars 12.5, and with the help of Google Earth imagery and ArcGIS. So for the results, we tried to divide the sections of Capisaan Cave into 5. These are represented through these images. We have the section 1, section 2, section 3, section 4, and section 5. So since section 1 and 3 have already maps that are generated, we tried to generate the map of section 4 and 5. So this is the section of Sabrina Cave, which is section 4. And this is for the Heaven Cave, which is section 5. And we also did cave entrance survey and limestone measurements. I will have a quick rundown here just to present you the 9 different entrances of the cave system. So this is the Lion entrance. This is the Sangat-Salug entrance and the Malukbo cave entrances, and the Lion closed entrance, the Lion main cave entrance, and the Heaven cave entrance, and Sabrina cave entrance. So for the cave sensitivity, we tried to look at different parameters to describe its sensitivity. And first, we tried to identify those plants that are present at the cave entrances. So this is the method that we did. We used the measurement of the entrance up to the twilight zone for the identification of plants. So all in all, we identified 91 plant species from 72 genera and 47 families. And from this, the family of aroid recorded the highest number of species, while the figs were the most represented genus. And there are also a considerable number of trees and treelets recorded in the area. Interestingly, the majority of the plants found in these cave openings were native, and 10 were endemic to the country. And there are also 5 vulnerable plant species that were identified. And these native and endemic species are presented in these pictures. We have the Rungia philippinensis, the Hoya camporifolia, and the vulnerable species of Alocasia nicolitziana, Asplenium vitiforme, and Begonia oxysperma. We have also Amalanthus macrodenus and Ringoglossum spumosum and Tuna calantas. Another is the Ficus odorata, Tepilianthus tenupes, and Podocarpus rumpi, Pneumatopteris levis, and Lia acuminatissima. So the canopy was generally open in the adjacent natural vegetation of the different cave openings. And there are also abundance of tree seedlings and of the gymnosperm almasiga and malakawayan in the area. But a very significant observation is that many parts of the karst forest were being cleared for plantation of gawid or the piper beetle, which is then perhaps the biggest threat to the survival of these native plants and the conservation of forest over limestone in this area. So this is typical scenario of how those different piper beetles were established in the area. So for cave fauna, we tried to identify the different taxa of snails and clams, but we were only able to identify 3, which is the Carbicula flaminea, which is in abundance in the 2 main entrances of Capisaan, which is in Malukbo and 1 and 2 caves. And also we have identified a sample of Alopecia species and giant African snail, which is, for me, it's a sign of disturbance. And for the arthropods, we did the belt transect method in combination with opportunistic sampling to identify the species diversity, evenness, and dominance using statistical software, including the relative abundance and relative frequency of species. So all in all, for arthropods, we were able to identify 24 species from 22 families and 12 orders, which is the order Araniae has the highest number of species recorded, followed by Opiliones, Orthoptera, and Decapoda. So just have a quick rundown of slides here again, just to show you those special features of this fauna, of these arthropods. So in section 1, we were able to identify or count 351 individuals of arthropods. And this is the picture of blind shrimp, which is very interesting since we were able to identify the genus of this as the genus Paricia, which is a very rare genus for blind shrimps. And hopefully this could be maybe another new species or site record for this genus Paricia. And we have the camel cricket, the spider crickets. And also we have for section 2, there are 246. So we have again spiders and Scutigoremorpha or Scutigera, the cave centipede, and the whip scorpion. For section 3, we have 466. This passage is very interesting because it is a home for this cave tarantula under genus Flagellus, which is still being identified if it is a new site record of existing species or maybe a new species. And also this huntsman and the Sundatal Pusa Quirino. So for section 4 and section 5, we have another special, very good identification of a cave harvestman, which is this one under species Dibunus. And this is, most of the Dibunus species is currently found mainly in the Philippines and some parts of the Southeast Asian nation, but maybe I think only 4 from the other country, but 7 in the Philippines. So for the diversity and dominance of these different cave arthropods, we can see here that the richness is quite high. The diversity was quite low since it is expected of these very special ecosystems because of those very limitations that they get from the cave. But dominance and evenness is quite even. So for fishes, we were able to identify 5, and these are the orange carp. We have the mudfish, carp, common carp, and the freshwater catfish. And for herpetofauna, we were able to identify 4 species of reptiles and 3 anurans, which are presented in the following pictures. We have the Cerodactylus philippinicus, Parvosincus decipiens, Eutropis multifasciata, a species of Platymantis, Calola rigida. We have the another species of Linonectes, and we have encountered one snake, which is the Oxyrhabdium modestum. For bats, we were able to identify 3 families under Rhinolopidae, Hipposideridae, and Miniopteridae, and, uh, and 4 species of these bats, which are the Rhinolopus arquatus, Hipposiderus diadema, Miniopterus australis, and Miniopterus sliversii. For birds, we tried to survey the surface landscape of Capisaan Cave using the point transect distance sampling method, and we were able to identify 61 species of these birds belonging to 35 families, and 36 species of these 61 were recorded to be endemic to the Philippines, and out of these 36, 8 species were categorized as threatened by the IUCN or the DNR. So the most abundant avian species in the sampling site includes the gray-faced buzzard, ridge-top swiftlet, Philippine bulbul, gray-backed tailor bird, and the pygmy swiftlet. Take note that the gray-faced buzzard is migratory, so you cannot see them year-round. But at this time, until the first quarter of the year, we can see them in abundance in the area. So this is the different pictures. This is the gray-faced buzzard. We have here the ridge-top swiftlet, the Philippine bulbul, the gray-backed tailor bird, and the pygmy swiftlet. So for species diversity, dominance, and evenness, both sampling sites, since we have 2, were considered high in diversity of birds. For resident status, we can see that both sampling sites have high species diversity, but this is very interesting, this one, the different species of birds that are considered threatened. So this includes the ashy-breasted flycatcher, the blue-breasted flycatcher, gray-throated sunbird, Luzon hornbill, ridge-top swiftlet, the rufous cuckoo, Sierra Madre ground warbler, white-browed shama, and the white-fronted tit. So for those bird enthusiasts out there, you can come and see these birds in their personal appearance in Capisaan. So these are the different pictures. For the overall biota sensitivity, since we're using an index, the index is composed of 4 classifications. So 0, 1, 2, and 3. 3 being the most high, or the highest, is given to all the different passages of Capisaan with those following justification. For cave hydrology, we also investigated how the behavior of water is found in Capisaan. So by looking at those streams, pools, drips, sieves, and possible aquifer connection. So this is the index that we used in the study. So cave passages were divided into sections with a span of 10 meters where these different hydrological characteristics were described. And what we did is that in this, for example, section 1, we tried to graph the index scores taken by these different partitions. So this would look like as the hydrology signature of section 1, for example. And with this, we were able to compute the total index score of the section, which is for section 1, we have 2.43. And these are the different photo captures in this passage. For section 2, we have 1.92 as index score. Section 3, 2.30. Section 4, 0.11. And section 5, 0.67. And for speleothem, this is the index that we used following the same partitioning for the different passages. And these are the results. For section 1, we have 2.40. Section 2, 1.82. Section 3, 1.35. And section 4, 2.56. And the same for section 5. For mineralogy, we tried to identify some enrichment deposition of minerals. Of course, other than calcite forming in the area. And what we tried to search are those presence of anthodites, gypsum flowers, and maybe some noticeable mineral veins that would indicate presence of other minerals. And these are the different coral fossils that are found in the limestone rocks of Capisaan, indicating that this area is mainly composed of limestone. So we were not able to identify any of those different indicators of minerals in the area aside from calcite. So it was given a score of 0. So for paleontology, we also look at the identified some maybe forms of fossilized remnants in the area of animals that are embedded in the walls, ceilings, and floors. And we were able to see only those coral fossils, which we tried to eliminate since it is a limestone cave. So having that kind of that result, we were able to identify or give a score of 0 for this cave paleontology. And for cultural, we tried to look at the presence of artifacts, ecofacts, and primitive artworks in the area. But since based from actual visit and literature reviews and interview from key informants, we were able to identify no presence of these cultural artifacts, so given a sensitivity score of 0. So for cave sensitivity, relative sensitivity, we tried to use the sensitivity scale presented by Harley et al. In 2011 from their research in different cave systems in Florida, using these different scores with different varying degrees of sensitivity. So for Capisaan, for the relative sensitivity score of all the passages, we were able to arrive at 0.37, which is slightly sensitive. This is influenced mainly because of these many 0 index scores from mineralogy, paleontology, and historic cultural parameters. But if we try to eliminate all those 0 scores, we can see from the result that from the relative sensitivity of slightly sensitive, it becomes severely sensitive. And also look at the noticeable change from section 1, which is now classified as critically sensitive. So for cave disturbance, so many parameters also were investigated. So first we have garbage, and this is the garbage index used in the study, following the same divisions or sectioning of the different cave systems or the cave passages. So for section 1, this is the garbage signature or trash. We were able to compute the index score to 0.51 for section 2.02, section 3.99, section 4.78, and section 5.22. And we also look at the damage to the speleothems found in the area. So we can see that using this index, section 1 gained a score of 0.30, section 2.60, section 3.41, section 4 2.25, and for section 5.44. And we also look at the presence of graffiti or the modern day inscriptions inside the cave, particularly on the cave walls and entrances, using this graffiti index. And the results showed that for section 1, we were not able to find any, so it's given a 0 score. But for section 2, there is a considerable presence of this graffiti, so we were able to give a score of 0.32. For section 3, also 0 for no trace. Section 4, 1.78, and for section 5, 0.22. We also checked for floor disturbance or the severity of sediment compaction in general foot traffic inside the cave using this index. So for section 1, the score is 0.89. Section 2.69, section 3.88, section 4.89, and section 5.78. We also look at the destruction of cultural artifacts and fossils, but since these are not present in the area, so this parameter is excluded in the computation. And we also look at cave sedimentation or the sediment deposition inside the cave, which is induced by anthropogenic activities. So we tried to characterize this through the identification of its origin, land use, and land cover change in the area, looking at the hydrologic behavior, and even interview of local cavers who've been entering the cave in the past. So using this index, and the results for section 1, the score is 0.25. Section 2 is 0.44. Section 3, 1.99. And section 4 and 5 were not included since there's no presence of active water flow in these sections. Okay, so we also tried to investigate on the species richness and population density percent decline in the area. But since this data that we have is a baseline or the first of its kind to be generated for species richness, what we did is that we tried to conduct a KII and focus group discussion in the area to just to have an idea of how this population density of various wildlife has declined. And the community using this index gave a score for the different sections. For section 1, 2.25, section 2, 2.25, so it's quite high. Section 3, 1.5, and 4, 1.33, and section 5, 1.66. So another parameter that we tried to look at is quarry mining, which is based on the effects of past quarries or presence of ongoing quarry concessions in the area. But since these are not observed in the area, it was given a disturbance score of 0. And this one is an addition to those existing index, which is what we consider novel in this research, the inclusion of regulatory protection, which is looking at the existing regulations and management plans that are institutionalized pertaining to the sustainable use of the cave resources. So using this regulatory protection index. So with those different evidences or observations, presented here, we were able to give the regulatory index score for Capisaan to equivalent to 1. And for another inclusion is the cave education, or since minimizing cave disturbance can be improved when the public is well aware of its fragility. So education activities that raise public awareness were measured as one of the indicator using this cave education index. So for the findings, given this different observation, we were able to give a score of or rating of 1 for Capisaan. This one is also novel since we tried to include the land use and land cover changes in the karst landscape of Capisaan using GIS and the FragStats, which is a fragmentation measurement software. And we used satellite images from 2001 to 2019 to compare how this land use and land cover has changed in Capisaan. So to do this, we did some ground truthing and geotagging in the karst landscape of Capisaan, as shown in this picture. And we also used this index to classify the gravity of the different variables that describes deforestation, agriculture, clearing, and build up in the area. So the result is interesting. So just a quick rundown how this changes from 2001 to 2019 using this 10 10 land classes, land cover classes. And just to appreciate how these changes have occurred in the area, we tried to group them into 3: forest, shrubland, and orchard, and agriculture and clearing with the different color indications. And this is what happened in the area. So there. So as a summary for fragmentation, the different FragStat metrics used in the study gave a final score of 1.33 for the index. For the relative cave disturbance score of Capisaan, by aggregating all those scores taken from or gathered from the different parameters, we used this disturbance classification used also by Harley in 2011 with those different degrees of disturbance. So this table shows that the mean disturbance score of the Capisaan Cave System is 0.28, which is grouped under minor disturbance. We did also some focus group discussion and scenario development in the area to get the aspirations of the community with regards to the ecotourism development in their area through, scenario development activities using two approach, which is the predictive approach and the normative approach. And we tried to we tried to investigate how the community would see these different ecotourism approaches in the area using number 1, business as usual, or the multi-stakeholder approach, or the second is the privatized ecotourism, public-private partnership, community-based, the fourth one is community-based ecotourism. So from those discussions, we were able to give general considerations in the ecotourism development of Capisaan. First is that the development and strengthening of the participation of the community to ecotourism must be reinforced through interdisciplinary program that co-opts the principles of sustainable tourism and natural resource management. it is also important to include the various elements of socio-ecological system in the development of ecotourism in the area that would balance the diversity of interests and positions of the stakeholders. And there should be a diversification of alternative livelihood, as well as involving the community in the conduct of researches, planning and implementation program, and decision making, which is already being done in the area. And we should remember also that community engagement in ecotourism entails a realization that the Capisaan Cave System is part of their well-being. So for the conclusion and recommendation, the zonal travel cost method technique is very applicable for those cave systems that have one-time individual visitation rates. And we were able to identify or provide an estimate of the recreational value of Capisaan and its total access value shown here. And for the vulnerability or sensitivity levels of Capisaan, the study successfully tested the applicability of the existing standardized cave sensitivity assessment index to tropical caves. And since this index built on the work of Harley et al., modifications were done by partitioning the cave passages and subjecting it to the evaluation using the index, which gives a more accurate evaluation, which can pinpoint the exact location in the cave passages that are needing the most attention and strict protection. And also it gives a better visualization of the different characteristics of cave passages, showing or tabulating the exact locations in the cave passages where high specific resource sensitivity can be found. And significant finding also that clearing and creeping deforestation for the plantation of piper beetle is a significant threat to the conservation of the native plants in the area. And of course, the diversity in the area. For the surface and subsurface animals, we can see that there is an abundance of diversity and high endemicity that makes it an excellent candidate for critical habitat establishment. And the identified high sensitivity of speleothem resources and occurrence of fascinating species of cave-dwelling arthropods and endemic birds makes the ecotourism potential of the area even more viable. So for hydrology, it is observed that it is, has a very active hydrology in the area. And among all those sensitivity parameters, biota was found to be the most sensitive, followed by speleothems and hydrology resources. For disturbance level of Capisaan Cave System, this use of index through partition measurements gave more specific identification of the relative index scores of the area. Also, it would give exact location in the cave passages where high specific resource disturbance can be found. For we can see also some isolated indications of disturbance from passages open for tourism. The same with garbage for those can be found in localized areas, that is frequented locations frequented by people and where floodwaters and main water course deposits its sediments or trapped in cave formations during flooding. Speleothem damages were seen in all sections, and these are brought by illegal entries. And also contributory factor is cave tourism and damages caused by more frequent occurrence of flooding in the area. Deforestation and gawid farming were strongly believed to have affected the subsurface features of Capisaan. So graffiti was found also in areas open for tourism, and also in areas where forced and illegal entries has been done. And high sedimentations is brought mainly by flooding and those water that enters the cave with high sediment loading. Uh, the reduction of forest cover and gradual increase in agriculture, clearing and build up has changed the land cover in the area. For fragmentation, the landscape fragmentation is apparent in the karst landscape of Capisaan. For regulatory protection, it was found to have reached a significant level of legislative effort, but implementation and sanctions are still wanting. And also for cave education, means to educate tourists on responsible cave tourism is identified to be more than enough, but should focus more on the communication effort and monitoring for the actual adherence by the tourists and visitors. The local community was identified to have rather low in scientific knowledge in cave interpretation skills, and also the skill of local tourism providers are the interpretation skills of local tourism providers are rather limited. But overall, the cave system in its designated sections were all determined to have sustained minor disturbances. And among those disturbance parameters, biota population, density, and richness decline and forest fragmentation were the top disturbances in Capisaan. So for the development of localized cave sensitivity and disturbance index, the data captured here provided a benchmark information needed in monitoring the socio-ecological condition of the cave. And these tools is recommended for managers and decision makers to scientifically prioritize actions to arrest or mitigate cave disturbances and protect and rehabilitate the significant physical cave resources in the area, and leading to the enhancement of the socio-ecological condition and delivery of ecosystem services of Capisaan Caves. So with all those results, we give these following recommendations that valuation studies to other ecosystem services should be continued, and recreation values of Capisaan Cave, the recreation value of the derived recreation value of Capisaan must be included in the economic projections and cost-benefit analysis of preserving or converting portions of Capisaan Cave System. And also to those significant biodiversity parameters that we were able to identify makes it Capisaan as an excellent candidate for critical habitat establishment. And the alarming extensive development of Piper beetle plantation should be controlled. And another is information, education, and communication must be done more intensively to schools and neighboring communities of Capisaan Cave to increase the cave education level for Capisaan. And for the last 5, we propose that the limit of acceptable change is recommended for determining the level of disturbance that is acceptable for Capisaan. And surface runoff and related disturbances can be minimized by constructing natural engineering and soil and water conservation measures in the area. And subsurface tourism-induced disturbances can be reduced by designating tight trails and preventing cave visitors to wander during those guided tours in Capisaan. And that the results also of the scenario development activities is reiterated for consideration by the concerned LGU. And the last, that any project or any significant project development in Capisaan must be done upon proper environmental impact assessment, and environmental management and monitoring plans are in place. So with all those activities, we would like to acknowledge the help of the United States Agency for International Development and the DAI, the Commission on Higher Education, and UPLB for allowing this to push through and giving all the resources for this to become a reality. So thank you very much for listening, and I think we're now ready for the open forum, Ma'am Banji.
Thank you also, Jason. And that's a lot of information. Maraming salamat sa iyo. At this point, we now invite everyone to the open forum. But before that, please allow me to acknowledge the presence of our partners in Protect Wildlife Project, the Philippine Biodiversity and Conservation Foundation, and Conservation International Philippines, or CI. So for the open forum, I will now turn over to my colleague, Ms. Rona Villanueva. Rona.
Yes, thank you, Ms. Banji. Good morning po sa lahat. I'm Rona Villanueva, the University Engagement Specialist of Protect Wildlife. For the open forum, we'd like to remind everyone to use the Zoom chat or use the raise hand button if you have questions for today's presenters. We already have several questions from our Zoom participants and FB viewers. So for the first question, let me read from Ray Thomas Kabigting. Hi, my question is, this is for Inigo. Yes, for Inigo. Do we have any assessment or data on the historic range versus current range of these species that we can compare to the rate of deforestation and land use change in their identified habitats? I think these are valuable information for the DNR to have in order to develop forest management programs geared towards restoring the historic range of these species, or in turn restoring their natural habitats. Inigo?
Yes, ma'am. Well, as far as I know, all we have is data on we have historical data on where they are found, but to know their present distribution, we have to revisit. I think we have to revisit all sites. We still have to revisit all sites. So basically, we don't have that data.
Okay, thank you, Inigo. Another question for you from Kim. Do pit vipers with different morphology have different venom effect? Was this covered in your study?
I think if we're referring to different morphology as different species, I think so. Kasi, well, according to a lot of studies, venom toxicity varies between from species to species. But between Tremerosaurus flavomaculatus of different morphs, well, maybe we don't have that data, and as far as I know, we don't have data on the actual. We don't have toxicity data on Tremerosaurus flavomaculatus.
Yes, thank you. Maybe that's for further research when you do your PhD or other research endeavors in the future. So this question from our FB listeners or FB viewers, from Mr. Jonathan Liwanes Mayuga. What is the this is also for Inigo. What is the conservation status of the pit viper?
So the Philippine pit viper is listed as least concern as of now by the IUCN.
Okay, IUCN classification. Let's go to one question for Mr. Dr. Jason Carranza. Sorry. The DNR clarified cave classification to determine appropriate use and management of caves and its resources in the country. Your research is comprehensive and commendable. Based on your findings, how will you classify the Capisaan Cave according to the three classifications issued by the DNR? Or is the Capisaan Cave System a mix of these classifications?
Yes, thank you, ma'am, for that question. Actually, three passages of the Capisaan Cave System has already been classified by the DNR, which is two have been classified as class 1 and one is class 2. So if I'm going to classify it again. It would be a mix of those different classifications. And I think we should stick with those classifications. Having two, I think we were able to identify naman kung alin yung dalawang passages na yon that are considered as class 1 and class 2, which is already often for guided tours. So we need to stick with that classification na rin siguro. Since itong findings naman natin does not contradict any of those classification, but rather it provides, kumbaga, if it is a picture, it provides a finer resolution of those cave characteristics that can be used in further identification of management priorities for those cave systems or for those different cave passages. Actually, this classification is intended for caves that are having long passages because luckily ang swerte nga ng Capisaan because we have the short passages which is around 100 meters only and those passages that are about 1 kilometer or more than 1 kilometer in total length. So we can compare and check how this index behave from these different scores. That's why I have mentioned that different cave passages have different cave management requirement based on those different parameters used. So yun ma'am. So it's a mix of classification.
Okay, thank you, Jason, for that clarification. Another question for Mr. Inigo. This is from our FB viewers. From Mr. Godfrey Jacusalem. Visayan region are more closer to Mindanao fauna region. How about Tawi-Tawi? Have you conducted collections or studies in Tawi-Tawi?
Yes. Hello, Sir Tagodo. Musta? So recently, I joined Sir Godo and their team. We visited Tawi-Tawi. Well, unfortunately, we didn't find any Philippine pit vipers, Trimerosaurus flaboplatus. But I'm guessing I don't want to exclude the other paik systems, so the other island groups. So to say that we only have two species between Luzon and Mindanao, ayoko pong sabihin ganun. So we're still open to the possibility na baka yung Visayas, ibang species din. So Negros Panay, ibang species din. Ganun po. So like yung Palawan and yung Batanes. So possibly yung Tawi-Tawi, if ever we find any, we have to look into it. Kung same species ba siya sa Flavomoclatus or ayan.
Yes, another future study for pit vipers. Inigo. So another question from Mr. Jonathan Mayuga. Are pit vipers being targeted by wildlife traffickers? Actually, if this is a question for BMB, but if you can answer this, yes, please do.
That ma'am, I'm not so sure. Pero I think I've seen photos posted online. So some are selling the pit viper. Ayan. Yun po.
Maybe I can, if the attorney Edward Lorenzo is here, can you share information on pit viper trafficking?
Morning, everyone. For a lot of posts a lot of posts on Facebook right now. For the most part, they're being billed as pests. In terms of trafficking, I'm sure there are going to be some collectors who are going to be. I'm sure there are going to be some collectors who will be who will be gathering some of these pit vipers. But for the most part, there's really not too much information on this particular species based on our records.
Thank you, Attorney Dong. Question for Dr. Jason Carranza. From Michelle Costales. Sir Jason, do you think the carrying capacity for Capisaan caves set at 100 packs daily is contributing to significant or any form of disturbance already? Or do you think the figure is very conservative or small? Please enlighten us if this was covered in your study too.
Yes,
Thanks and congrats again.
Yes, ma'am. Thank you, Ma'am Marichel, for that wonderful question. there are a lot of answers to this question, and it could be derived from the experiences of the cave guides who are touring inside the cave system. Uh, the computation of 100 packs daily is based on the time interval that is needed to accommodate all those tourists in different setting as they go inside the cave. The problem here is that once they are inside the cave, I think nagkakaroon na sila doon ng closer gaps or they even merge inside. So this has a lot of effects already in the management of tourism inside the cave since the number of cave guides that could cover this number of tourists is 1:5 already, that is already present 1:5. But then once they merge, it is very difficult to contain them. So the problem here now is that we need to find a way really to help or uh, ipaunawa natin sa mga cave guide that they should become strict in maintaining the interval inside the cave. That would be more manageable pagdating sa loob. And for the further details of the carrying capacity, this can be done by another research, which is not covered by my study. But then I have proposed that the limit of acceptable change is will be implemented for or will be computed for Capisaan since every time a cave is entered, there is damages or disturbance that is being done. So we need to identify hanggang saan ba yung gusto nating damages na pwedeng that we can say that it is tolerable for Capisaan Cave. So yun yung mga magiging monitoring factors natin later on, whether if the number that we allowed to enter, the number of tourists that we allowed to enter inside the cave is within the range of acceptable change that we want, or medyo nagbago na or it goes beyond the range that we want, then that's the time that we should try to reconsider or recomputing how many tourists can enter really those different cave passages. So my suggestion is that we should conduct another study using different carrying capacity limits in the area. Since meron tayong different carrying capacity measurement like physical carrying capacity, how the facilities in the area can accommodate all the number of tourists that are coming. We have the biological carrying capacity and many other carrying capacity limits that we can study and merge the different results to come up with a better number of tourists and managing how they will enter or stay in the area. I think yun ma'am, yung sagot ko po dun.
Thank you, Jason, for that answer. So this is another question for follow-up question for Jason
Yes,
from our FB viewer. For Dr. Carranza, gawad farming is a way of life for many people in communities around the cave system. What is your recommendation to reduce or cushion its impact on the cave system and its biodiversity? Question mark.
Pardon mo, ano po yun? Gawad farming?
Gawid. The Piper beetle?
Gawid. Sorry ma'am.
Gawid. Sorry.
Gawid. Yeah, that's it's apparent that the plantation is growing in the area. So, but then there are mitigating measures that being implemented by the DNR and the provincial government already. Pero there's a parang test of powers eh. Kung sino yung mas nangingibabaw, is the government or the people who are in need of livelihood in order to survive. So we need to, if we want to limit how the gawid farming is being practiced in the area, we need to provide them a good alternative. Or ang nasa isip ko nga, how are we going to marry these two land use? Are we going to protect the forest that is protecting the cave system, or are we going to allow the farming system that gives livelihood to the people na hindi naman siya makakasira dun sa cave system? So we need to find a ground kung saan sila magmi-meet sana. And that ground is not yet set, I think. So we need to study this one. Kasi parang nagkakaroon ng agawan which is dapat na gawin na sa lugar, protection or conversion of land. But then for us, since we are in environmental science and in sa DNR, as much as we want to preserve the integrity of the forest that protects the cave system. Because once the cave system is lost or damaged, it's very difficult or even impossible to bring it back to its original condition. But the beauty of this is that even some parts of the cave system has been already damaged. Pag nandun ka, nakita mo, it still gives the feeling of home. Kahit na damage na siya, andun pa rin yung awesomeness of that creation or resource. But then we just don't want to have all those resources change into that kind of setting. So, kailangan there's a middle ground kung saan both development and protection should be favored.
Yes, our priority, of course, is for biodiversity conservation, but we have to find the balance between the needs of the community and for the balance between livelihood and then the biodiversity conservation.
Yes,
Yes, thank you, Jason. This another question to Inigo. Have you checked whether your samples from your study sites were previously collected by earlier taxonomists and were previously named or synonyms other than T. flavomaculatus, or will it affect your taxonomic alternative in the future? I guess this has been discussed in your related lists, previous studies, right?
Yes, ma'am. So for specimens from Mindanao, so pit vipers from Mindanao were originally described as Trimerosaurus schadenbergii by Fisher, I think in 1885. So if we were to prove that Trimerosaurus flavomaculatus of Mindanao are indeed a cryptic species or a separate species, I think we would have to use the name Trimerosaurus schadenbergii. So yun yung future niya, if ever ma-prove.
Yes, maraming future studies for both pit viper and Capisaan Cave System. There is a follow-up question for you, Inigo. Following the question regarding the pit viper's venom, what are its effects? Can one die after being bit? And is there already an existing anti-venom?
So, well, according to studies, ang venom ng pit viper ay hemotoxic. So it attacks the blood vessels, the heart. Yeah. Well, although reports or published literature on Philippine pit viper bites are very rare, I think mostly according to locals, the effects include intense swelling, so mamaga. Kung saan sila nakagat, namamaga yung buong, nakagat sa finger, namamaga yung buong arm. Followed by extreme pain and then necrosis, so nabubulok po. Ayun. Pero I think reports of deaths are very rare. And as far as I know, they're not considered deadly. They're dangerously venomous but not deadly.
Yes, if I tama yung recollection ko, hindi naman sila nang a-attack, di ba? As much as possible, they avoid the people. Kaya rare yung or bihira ang nakakagat ng mga pit vipers. Tama ba ako?
Opo. Unfortunately, so since ambush predators sila, magaling yung camouflage.
Yes.
And then, so they're sit and wait predators. So ang tawag nga ng iba sa kanila, ahas tulog, since hindi talaga sila gumagalaw. And minsan mahirap talaga sila mapansin. And unfortunately, yun nga po, may mga reports na, especially yung local hunters, So yung, for example, yung mga from tribes ng Agta na nag-hunt ng shrimp and eels at night sa streams, rivers. And these are pit viper habitats. And minsan natatapakan nila. And ayun, so ang unang defense ng pit viper, kakagatin sila.
Okay.
And also yung regarding antivenom, wala po tayong antivenom. So out of the so we have around 150 species of snakes in the Philippines. 33 of these are considered dangerously venomous, including yung marine snakes. And sa 33 na to, only one species nagpo-produce tayo ng antivenom. This is the Philippine cobra. So for the pit vipers, wala po tayong antivenom.
Yes, so species specific ba yung anti-venom?
Dapat ma'am, species specific. Kasi according to studies, venom toxicity and effects varies between species and species. Between species.
Okay, thank you, Inigo, for that. There's a follow-up question from Godo. I don't know if this is a question or a comment. Maybe you should join us here in West Visayas fieldwork for the pit viper. Is there a taxonomic system like bird to bias criterion, just looking at morphology without DNA?
Without DNA. Unfortunately for reptiles, magulo po kasi sila kasi again, we have cryptic species that look alike. And so recent trends on reptiles have been using molecular techniques talaga for delimiting species boundaries. And Sir Godo, sasama ako sa inyo sa Visayas.
Okay yan, sasama na daw si Inigo. Okay, maraming pong salamat sa mga participants from this Zoom room and from our FB viewers, and also for our two speakers, Dr. Jason Carranza and Mr. Inigo Del Prado. At this point, we are concluding the second episode of Connected to the Wild Biodiversity Research Series. Before you leave, please don't forget to fill out our feedback form from that posted on the Zoom chat. Also, we invite everyone to our upcoming third episode featuring our other research studies on maybe third week of October. We'll post details on the third episode on Facebook. Okay, muli po, maraming salamat and see you next time.
Thank you, ma'am.
Thank you, Inigo, and thank you, Jason.
Thank you.
Thank you, ma'am.